Everyone, this is lecture 14, the molecular basis of inheritance. In other words, just DNA. So again, this ties into the concepts that we've been covering about meiosis, Mendelian genetics, and non-Mendelian genetics, because DNA is what makes up chromosomes, as we kind of touched on last lecture. So how do you make more DNA when you need to make new cells? Either for mitosis or meiosis, you need to make a copy of DNA, right? In the S phase of interphase. So DNA replication is the process that does this. You get the DNA that you have, you make a copy, and boom, now you have the sister chromatids. So DNA replication is what allows genetic information to be inherited from a parent cell to the daughter cells by mitosis, and from generation to generation, starting with meiosis. So how does that happen? So here is an unduplicated chromosome, and in this purple figure of it, it's kind of like zoomed out. It just looks like a string. But if we were to zoom in, we would see that DNA double-stranded helix that we kind of picture as DNA. But again, it's just like a long string, so that's why it's organized into chromosomes, chromatin, and then chromosomes. And so here's an unduplicated chromosome, one DNA molecule, and then the proteins that it's wrapped around. And so if we zoom in, here is one DNA segment from a chromosome, and that carries one gene. And so each gene, again, is a unit of hereditary information consisting of a specific DNA sequence. And so when replication happens, DNA replication begins by kind of like unzipping the DNA strand, and then using each half to start building the new half. And then at the end, there are two DNA molecules that get distributed to the daughter cells. And so DNA replication actually happens at multiple sites, but here's just zooming in on one part. So you're unzipping, building new strands. And so after you finish duplicating the whole strand, because again, it's very long, at the very end, you end up with two DNA molecules. And then when they get condensed, they look like chromosomes. And that's the two sister chromatids, how you've made two sister chromatids. They are duplicated and then condensed into these chromosomes. So how did we get there? We could kind of keep going with this story of, Mendel discovered something gets passed on. Some other people discovered that there are chromosomes that get passed on. And here we kind of continue this mystery. So early in the 20th century, the identification of the molecules of inheritance, posed a major challenge to biologists. They saw something gets passed on from parent to offspring. But what kind of molecule is it? So there is a group led by Thomas Hunt Morgan, so T.H. Morgan. His group showed that genes are located on chromosomes, and so they had to figure out, okay, chromosomes, what is it that's part of chromosomes that gets passed on? Because it's both DNA and protein that make up chromosomes, right? So which one of those two is it? Because they're both in chromosomes, and we know that chromosomes get passed on, but is it DNA or is it protein that is like the genetic material? So the role of DNA in heredity was first discovered by studying bacteria and viruses that infected them. So now on to another group that made the next series of experiments and found, okay, it's DNA that is the important part of chromosomes that gets carried on. So the discovery of the genetic role of DNA began with research by Frederick Griffith in 1928. And so this guy Griffith worked with two different strains of bacterium, one pathogenic and one harmless. So it was kind of the same species, but there's different strains. There's different possible strains. And so it's like subspecies, basically. But when you talk about bacteria, you refer to them as strains. So there was one that I'm just going to show as this little cartoon here is pathogenic because it's like thicker. And then the other one will show as harmless. There's going to be a real figure, but I thought this might help simplify it a little bit. So there's these two different strains. One pathogenic, meaning it can make people sick, make organisms sick and die. And then the other one happens to be harmless. And it's like, it's not a totally different bacteria species. It's the same species, just different subspecies, different strains. So he was studying, okay, what is it that makes the difference between the pathogenic one and the harmless one? And so he found that if he took the pathogenic strain and he basically cooked them and heat killed them, if you bring it to a high enough temperature, that just, denatures all the proteins. And so it killed the bacteria of the pathogenic strain. And he was like, well, what if I mix all this dead stuff of this pathogenic strain and I mixed it with the living cells of the harmless strain? What happens? Does anything happen? And so he found that when he did this, some of the living cells ended up becoming pathogenic. And so he was like, what the heck? Something got passed on. And it didn't even have to be from a living thing to a living thing. It was from a dead thing to this living thing. So something in that got transferred is important. So yeah, when he heat killed the remains of the pathogenic strain, and he mixed it with the living cells of the harmless strain, all of a sudden this harmless strain became pathogenic. And so he called this phenomenon transformation, because it's like it transformed this into this. And so this is still a term that we kind of use, and we actually do lab protocols where you can take DNA from other bacteria and put it into a new bacteria, and it'll take it up and express it, and that's called bacterial transformation, because you're like transforming this bacteria. So he called this phenomenon transformation, now defined as a change in genotype and phenotype due to assimilation or taking in foreign DNA. And so here is the figure, from the textbook demonstrating this experiment. And so if you were to grow bacteria on a petri dish, on an agar plate, you can see differences in what the colonies look like of the bacteria. So colonies are like spots where thousands and thousands of cells of bacteria have replicated. And so there's enough bacteria there to be visibly observable. And so that's called a colony. And so sometimes the colonies can look different from each other. So sometimes if you look at it, the plate, the colony can look smooth. You don't even have to touch it. It just looks smooth. And then sometimes they look different. And so that's why he came up with this kind of naming scheme for these two different strains. So he called the smooth colonies the S cells. And then he called the rough colonies R cells. So it might get a little bit confusing, but that's why I wanted to put this to the side. So S means like smooth, and that's the pathogenic one. R means rough, and that's the non-pathogenic or the harmless ones, just like I kind of showed on the other one. So when he took those living S cells and put them into a mouse, the mouse dies. So yeah, the cells are pathogenic. They can cause disease and death. And when he put the living R cells into the mouse, for some reason, the mouse was healthy. That strain was harmless. It was non-pathogenic. And then so he killed some of the S cells, the pathogenic ones. And if he were to inject that into a mouse, because it was, effectively heat killed, the mouse was healthy. It's fine. But if he mixed the heat-killed S cells with the living R cells, then the mouse died. And so he was like, what is up with that? I know that the R cells don't on their own kill the mice. And I know I heat-killed these S cells because when I injected them, the mouse was healthy and it didn't die. So something got transferred from these dead S cells into the living R cells, and that made these cells different, and then the mouse died. So later on, if you were to look at the cells Now all of a sudden they are S, like their colonies look smooth. And so something got transferred and something got expressed. So like a new genotype and phenotype because of this new DNA. And again, at that time, he didn't know it was DNA, but they're trying to figure out something is passed on. So yeah, a genetic trait can be transferred between different bacterial strains. And now we know it's the DNA. So after that, later work by a different group, by Oswald Avery, Macklin, McCarty, and Colin McLeod, identified that substance, the transforming substance, as DNA. And so it's a lot of work to get to what we know now, right? But everybody had to start somewhere, and it was tackling like a different aspect of this question. And that is how science is done. So at this time, this group said, oh, the transforming substance is DNA. But many biologists were still skeptical because not that much was known about DNA at that time. Again, we all had to like build the knowledge from somewhere by doing these experiments. So they're like, okay, well, let's get to know a little bit more about this DNA and we'll see if this is really the transforming substance. So then there was more evidence that DNA is the genetic material from studies of viruses that infect bacteria. So viruses, I think we mentioned, they're not technically alive. They're just stuff. They're just particles, basically, and molecules. But bacteria are alive. They are prokaryotes. They have all the characteristics of life. But there are viruses that infect bacteria. And so those are called bacteriophages, or just phages for short. So those are viruses that infect bacteria specifically. And so a virus is DNA, sometimes RNA, enclosed, so closed in, by a protective coat, and often it's just protein. So I kind of think of, we show pictures of viruses as like a round thing, and then there's like the DNA or RNA inside, there's genetic material, and then there's usually stuff coming out. And those are like proteins that are on the surface. And so that's all that viruses are. There's no cell membrane because it's not a cell. There's no cytoplasm, nothing. It's just those two things, DNA or RNA, and then that protective coat. And that's usually protein. And that's all viruses are. And they're tiny, tiny, tiny. So yeah, even though we show them as like possibly a circle, or maybe it has like a weird shape, like a tetrahedron or whatever, keep in mind that they're tiny and they are not cells. So a virus is just those two things, the protective coat, usually protein, and then DNA or RNA inside. And so if we study these viruses and what that infects bacteria with, then that, taught us some more. So bacteriophages, or again, just phages, have been widely used as tools by researchers in molecular genetics because, yeah, they are genetics. They have DNA or RNA, even though they don't have everything else. Like, they can teach us some stuff. And so here is a picture of a virus, a bacteriophage, infecting a bacterial cell. So a bacteriophage can have different shapes, but a lot of times it's like this weird, it almost looks like a little, like a rocket or something, like it has this kind of like head. It has a tail, and then it almost looks like little legs. But again, this is just protein basically, and then the DNA inside. And then so it kind of like injects its DNA into the bacteria through this tail. Here's the head, the tail, and then it kind of like injects the material in there. Yeah. And so these are tiny, tiny, tiny. Like, bacteria are huge in comparison. So, in 1952, this group, Alfred Hershey and Martha Chase, showed that DNA is the genetic material of a phage known as T2. So a phage that, kind of looks like this. They were trying to figure out what is that genetic material inside. Is it DNA or is it RNA or what? And so they showed that DNA is the material inside of that phage known as T2. And then they designed an experiment showing that what is it that gets injected from the bacteriophage into the bacteria. And they showed that it is DNA. So they designed an experiment showing that only one of the two components that make up the T2 bacteriophage, there's just DNA and protein, one of those two enters the E. coli cell in this experiment during infection. And so they concluded that the DNA that got injected from the phage is what provides genetic information. And so here is a figure on the next slide. So again, bacteria phages, and like other viruses, they just have two components. They have protein or DNA or RNA inside. So here are two parts of the experiment that they did. So in batch 1, they labeled the protein with a radioactive tracker. So radioactive sulfur was used to track the protein. And so that would kind of show pink. And so if that is what, well, that's shown here in pink. And so if it's protein that gets injected into the bacteria, then that tracer is going to be in there. And then in batch 2, they labeled with a different radioactive isotope. They used phosphorus to mark DNA. And so if this marks DNA and DNA is what's getting injected, then that's what you'll see on the inside of the bacteria that came from the phage. And so on the top half, they're seeing protein, and then on the bottom half, they're seeing is it DNA. And so on the top half, this is what would happen if this was the case. So if the phage was injecting protein in here, then you would see protein inside of the cell. And then if you were to take all these bacteria cells that got infected and put them in a test tube and centrifuge them and spin them really fast, that pulls different things to the bottom at different rates. So things that are heavier are going to get pulled to the very bottom, and then things that are lighter are going to stay at different levels. So this helps separate parts of the cell. So yeah, in the test tube, when they spin it, they would expect to see whatever's important at certain parts of the cell. And so the fact that they didn't see any of that protein inside of the cell means that's not what got injected. Yeah. And then so in batch 2 on the bottom, this is what happened. They found that the DNA that was marked by the radioactive phosphorus did end up at the bottom with all the rest of the parts of the cell. So that must be what got into the cell. And so that's what they, determined. Oh, okay, so something gets injected from the bacteriophage into the bacteria. And it's not the protein because the protein stayed in the liquidy parts instead of being inside of and stuck with all the cells. And so when they looked and they saw that radioactive phosphorus at the bottom in the pellet, the clump basically is what we call a pellet. So what they found in the pellet was actually the DNA. And so that tells them that's what got inside of the cells. So now we know even more about DNA. We won't go into how they found this out, but yeah, now we know even more detail about DNA. So DNA is a polymer. So remember, it's a large molecule built from the smaller molecules called monomers. So if you need a review of this, you might want to go back to chapter 5. So DNA is a polymer of nucleotides. And so one nucleotide is one monomer, and then you stick another nucleotide and build the polymer of nucleic acids this way. But even a monomer can have different parts, right? So a monomer of a nucleotide is made from these things. So there is a nitrogenous base shown here in green. And that can be different ones, which I'll get into in a second. So that's one part. There is a sugar. In the case of DNA, the sugar is deoxyribose. And then in the case of RNA, which is also a nucleic acid, it's ribose. So again, if you wanted a reminder of all that stuff, you might want to go back to chapter 5. So that is a sugar and then a phosphate group. You've heard all about phosphate groups by now, right? So that makes a nucleotide is those three things: a nitrogenous base, a sugar, and then a phosphate group. And then, yeah, in the case of DNA, it is specifically deoxyribose as the sugar. And so the nitrogenous base that is in this position can be different ones. There can be adenine, thymine, guanine, and cytosine. And so, yeah, this is one nucleotide made of those things. And then I will show another picture of the other nitrogenous bases. But yeah, you can kind of plug in different ones here. And those make different nucleotides. And so we shorten the names as adenine, just A, thymine T, guanine G, and then cytosine C. And so another important experiment is the one done by this guy, Chargaff. So in 1950, Erwin Chargaff reported that DNA composition, so those bases, A, T, G, C, varies from one species to the next. So it's not that every single species uses DNA, but there's different sequences in each species. Like there's different amounts of the A's and T's and G's and C's in different species. We don't all have the exact same sequence. Otherwise, we would have the exact same makeup of all those letters, all those nucleotides. So this evidence of molecular diversity among organisms made DNA a more credible candidate for the genetic material. The fact that basically all living things use DNA, but then that the DNA also is different between the different species makes sense. And so that helped them think, okay, yeah, maybe this DNA stuff, there's something to it, So he found two things. These two findings became known as Chargaff's rules. So one of those things is how the base composition of DNA varies between species. So yeah, different species are going to have different sequences of A, T, G, C. And then his second finding is that in any species, the number of A and T bases is always going to be equal. And then the number of G and C bases is always equal to each other. So if there is 33 A's, then there's going to be 33 T's. If there's 47 G's, there's going to be 47 C's. And that is for a reason that we know now. So the basis for these rules was not understood until the discovery of the double helix. But he, again, was onto something okay, if we look at the nucleotides, there's some correlation. There's always going to be this many number of A and T's equal, and then G's and C's are always going to be equal. So yeah, later on we figured out why. But that's Chargaff's rule. So the basis for these rules was not understood until the discovery of the double helix. So this is going back to that idea of how DNA is a polymer of nucleotides. And so this is one nucleotide. It has those three things: a nitrogenous base, a deoxyribose sugar, and then a phosphate group. And so in this nucleotide, the nitrogenous base that is here. And again, nitrogenous means that there's nitrogen. So the nitrogenous base that is here is adenine. This one is cytosine. This one is guanine. And then this one is thymine. And so we shorten them as just ATGC or ACGT. And so this is helping you see how they are slightly different from each other and consider different bases. So even though some of them look similar, like T and C, they look kind of similar shape-wise, but there are small differences. And so yeah, they're totally different nitrogenous bases. And so what Chargaff was saying is that as many A's as you find in a molecule, there's going to be that many T's. Like it always is equal. And then same with G's and C's. And so as we learned more about DNA as a molecule and its structure, we learned why. So after DNA was accepted as the genetic material, then the challenge was to determine how its structure accounts for its role in inheritance. So what is its structure? And how does that lead to it being passed on the way it does? So another group, Maurice Wilkins and Rosalind Franklin, were using a technique called X-ray crystallography to study molecular structure of different things. And so they happened to study this. And so X-ray crystallography takes something, you take a molecule and you kind of like freeze it in a crystal. And then, so say this is like DNA. You freeze it in a crystal and then you shoot X-rays at it in a fancy machine. And then the way the X-rays bounce off tells you something about that molecule. And so they did that with DNA. And so Rosalind Franklin produced a picture of the DNA molecule using this technique. And so this is Rosalind Franklin. And then this is her picture that she found, that she took of DNA, and that led her to, I don't understand this picture, but she did. And she found that the way that things got bounced off meant that this must be the molecule in this shape for the X-rays to bounce off this way. And so this was actually huge, and I'm going to talk her up because she doesn't really get enough credit for this. I mean, it's huge. Without this picture, they wouldn't have figured out more about it. So yeah, she's a personal, female in STEM hero. And so her X-ray crystallographic images of DNA are what allowed another group to deduce that DNA was helical. Like there were different groups working on this at the same time. And this other group happened to get her picture and we're like, oh, okay, this makes this then. So basically stole her data and got credit for it. And so, yeah. So Watson and Crick, I'm salty about it. They get all the credit, but it's because they had that figure that she took. So that allowed James Watson to say, oh, DNA is helical. And they also allowed him to deduce the width of the helix. So not just that it was like a helix, but also how wide it was, and then the spacing of the bases. So the pattern in the photo suggested that the DNA molecule was made up of 2 strands, actually. So it wasn't just one solid one like in this picture. It's 2 strands that are held together, forming a double helix. So Watson and Crick built models of a double helix to conform to those X-rays and chemistry of DNA. And then so when they built it, it kind of helped solidify, okay, from all this data, and we built this model, this seems like this is the case. Yeah. And again, that's based on what Rosalind Franklin figured out. So Franklin had concluded that there were 2 outer sugar phosphate backbones. So if we go back here, the sugar and phosphate that's alternating, that's what makes like the backbone of the helix that we picture. Yeah. So Franklin had concluded that there were two outer phosphate sugar backbones, and the nitrogenous bases paired across, and that's how it held the two together. So basically, she had it figured out too, is what I'm trying to say, but she doesn't get the credit. And then so Watson and Crick built models, and then Watson built a model in which the backbones were anti-parallel. So if this is one half of the strand, here's a highlighter. Okay, so here, I'm going to go based on the clip and the like point. So if this is going one direction, the other end of the, whatever. So the other end is going the opposite way. So if the writing part is on this side, on this strand, and then the writing part is on this strand going opposite, that's anti-parallel. Parallel would be like they're both going the exact same direction. So that they're going anti-parallel. Yeah, he did it like that, and then that's how kind of the math worked out. So this is a visual of DNA, and so here is one kind of like rung on this ladder, and so each side with the blue and like tan or whatever, that's alternating sugar phosphates from each nucleotide, and then they happen to twist a little bit just because the physics of how these atoms come together, like it has to twist. And so there are two twists actually. So there is one twist and then there's like another one. And so if you ever notice, there's actually like a wider gap in one twist and then a smaller twist and then a bigger twist and then a smaller twist, or like a smaller gap between twists. So this is what they call it, well, a turn. So there's one full turn for every 10 base pairs. And that's based on, again, the spacing and the physics of it, like how they stick out and interact with each other. So that's the sugar phosphate backbone. And then these are the bases that reach across and then connect to each other as the twisting happens. So there's one nucleotide connecting to the next, and then there's one strand connected to the other strand, and they are running anti-parallel like that. So at first, Watson and Crick thought the bases paired the similar ones together. So like maybe A goes with A, G goes with G. But that did not end up working out. It didn't result in the uniform width that would make this kind of turn possible. So instead, it turned out that pairing a purine with a pyrimidine, I'll talk about that in a second, resulted in a uniform width consistent with the X-ray data. So A and G are called purines, and then C and T are called pyrimidines. So purines have 2 rings, pyrimidines just have 1 ring. So yeah, you have to pair one with 2 rings with one 1 ring, and that's how it works out and is consistent with X-ray data. So if you were to put like a purine with a purine, it would be too wide, and then a pyrimidine and a pyrimidine is too narrow, so it has to be one of each. So either A and T or G and C for it to work out that way. So Watson and Crick reasoned that their pairing was more specific, dictated by the base structures. So it's not just any purine and any pyrimidine. It's that adenine paired only with thymine. So A always binds with T, and G always binds with C. Guanine only pairs with cytosine. And so that's how it has to work out. And so when they figured that out, that explained Chargaff's rule. So in any organism, the amount of A equals T because they only bind with each other. So if there's this many A's, there has to be this many T's because they only bind with each other. And then the same for G and C. The amount of G always equals the amount of C because they pair only with each other. So if you were to be given like a problem, you know, oh, the total amount of DNA adds up to 100%. If 33% of the DNA in this sample has A, then how much is T? That would also be 33% because they have to equal each other. And then, so that means that the rest is made of G and C, and then you have to divide that by half. Hopefully that makes sense. And so this shows why, like on a molecular basis, it has to work out this way. So adenine and thymine always pair together because that's what is compatible. Like what sticks out on adenine can bind with what sticks out here at thymine, and then same here. And so there are two hydrogen bonds that hold these pairs together. You know, there's one hydrogen bond and then a second hydrogen bond. So A and T are always bound together, and there's always going to be the two hydrogen bonds. And then for G and C, they always pair together, again, because of what sticks out on each one and what's compatible. And so between G and C, there's always going to be 1, 2, 3 hydrogen bonds between guanine and cytosine.